One of the things that we now know is that these targeted therapeutics are going after important mediators of normal cellular function, and that we have to understand how the particular targets that we have drugs for are abnormal in the cancer cell in such a way that we would have a therapeutic index between the cancer cell and the normal cell. What is coming out of this is that there are very strong homeostatic mechanisms present in normal cells that bring those cells back into equilibrium and allow them to survive the stress that occurs when you block important pathways. Cancer cells, in contrast, because they have many different aberrations and many different mutations, may have two events that make them vulnerable. The first one is that they become addicted to a particular signaling event. That is, they have a mutation in a gene, amplification of HER2, and in contrast to normal cells where that's one way in which those cells can function, the cancer cells are addicted to that event and they have no other alternatives but to die or stop growing when you block that pathway. The second possibility is that cancer cells, because they have lost their homeostatic mechanisms, are actually vulnerable to the effects of blocking important signaling pathways. They have fewer backup mechanisms, they have fewer survival mechanisms in place, and less robustness to challenge.